We are going to talk about karyotypes and how we know how many chromosomes in an organism we have. Nearly all human cells contain 46 chromosomes inside of their nuclei. Here we see photographic representations of all of the chromosomes in a cell from a male and all of the chromosomes in a cell from a female. This is what we call a karyotype. It's a photographic representation of the chromosomes in a cell. So this is one karyotype. It is from a human male. We see 46 chromosomes from the cell of this male. They're all represented here. And this is a karyotype of a human female. We also see 46 chromosomes from the body cell of this female. Chromosomes are, of course, not arranged like this in a cell. Here they are paired up perfectly. This is just in the karyotype. We put them like this on purpose and then photograph them. We pair them up before we take the photograph in order to make the karyotype. So, of course, they're not arranged in this organized manner in the cell. So, the definition of a karyotype is it's the arranged set of homologous chromosomes, which means similar chromosomes that represent each other, and it allows the detection of chromosomal errors using nucleated cells. So you use cells that have nuclei because these will have DNA in order to make a karyotype. And once you pair them up in the homologous pairs and you look at them in the karyotype, you can tell if we have chromosomal errors, if a chromosome is broken, for example. And you take these nucleated cells either from an individual or from the amniotic fluid of a pregnant female, which is the fluid in which the fetus is found.